I would like to express my sincerest gratitude to Professor Sharma for inviting me here. And it's a matter of immense pleasure that I'm sharing this stage here. I have been an alumni here. I did my bachelor's here in the Department of Genetics and Genomics. So I'm feeling amazing to be here. And I'll proceed to discuss my PhD research work with you. If you have any question, you can stop me or you can wait until I reach the end. So, can we go ahead? One more slide. Okay, so I have worked like for some 6-7 years before finishing this 5 years PhD program, and I have worked in several labs. So I will give you a brief insight about the research I have done in different laboratories before finishing my PhD. So I submitted my master's thesis in Indian Institute of Science Education and Research, Kolkata, and my master's thesis was on optimizing neuronal differentiation from human-born and jelly-derived mesenchymal stem cells. My goal in master's thesis was to optimize a protocol which was suitable for differentiating water jelly mesenchymal stem cells. These are the stem cells derived from the umbilical cord, and using different combinations of different concentration of phospholin and EFGF, and the cell number and incubation time, I got around 35%. It was like 1 year master thesis, so I got somewhat good percentage of cells differentiating into neuron-like cells, as you can see the morphology in the immune sample. And after submitting my master's thesis, I continued a 2 years research project within the ISA Kolkata, where my goal was to clone, express, and purify the surface exposed colonization proteins from Campylobacter jejuni for the and to check its differential host responses. The main aim of this project was to develop a vaccine against this bacterial zoonotic pathogen, Campylobacter jejuni, which mainly causes the intestinal bowel disorder and dysentery. So I did. It was a fresh project. I wrote the grant along with my professor, and I came up with conserved proteins which would be used for as a vaccine candidates, and I cloned them into the bacteria, expressed, purified. Later on, it was checked for the immunogenic responses in rabbits. And then I started looking for PhD, and meanwhile, while I was searching for PhD, I got a junior research fellowship at Jawaharlal Nehru University. And my goal there was to do the back purification of ECCA1 protein, which is a protein required for transferring the pathogenic proteins in the host cells by mycobacterium tuberculosis. And the ultimate goal of this project was to crystallize this protein. And crystallization field itself is very tricky, and nobody knows when you will get crystal and what you need to do. So luckily, I read the Nature's paper, and there it was in situ proteolysis, and I used a combination of different proteases. And in 5 years, first time I saw the crystal of this protein, and it was immense. It was an amazing feeling. But you are like, unless you get strong, very good interactions, you cannot be sure that you are going to get something or not. So these did not diffract very well. I got some 10 strong diffraction, and you don't see any helix or amino acid. So, but it was the start. Can you give me the next? And then I started my first year of PhD at the BenGurion University of Negev. And my first lab there was the structural biology lab, because my previous experience was in structural biology, and cardiovascular microscopy is still now a booming field. So my goal there was to do the membrane protein purification. It's very tough to purify proteins. And the good thing about cryo-electron microscopy is that even with the smallest amount of protein, you can continue doing imaging, and you can get a decent structure, which is not possible with the crystallography. Protein crystallography, you need a good amount of protein. So there the sequence of protocol is like you do membrane protein purification. Here in this case, it is PCG5A, a cholesterol transporter, and then you make the nanodisk. This is the second is the structure of nanodisk. It mimics the small membrane-bound transmembrane proteins, and there you put these phospholipids. Phospholipids go and surround the membrane protein, and they mask with the hydrophobic patches of the membrane protein. And MSP1T1 is another protein that circumvents this whole lipid structure and makes a small nanolipid structure, and then you purify using the size exclusion chromatography. Then I did negative scanning to check if the nanolipids they look amazing or not, they look good or not, and then we do this is fourth is the 2D classification. Using the tri-electron micrograph, you do 2D classification, 3D classification, and then you do this. So, but after doing 1 year of this research, I found that more than molecular biology, it's kind of repetitive work, and most of the work includes image processing and having a very strong computer science background. And then ultimately I decided to continue my journey in the molecular biology. Then I switched my lab and I joined plant molecular biology lab. So today I'm going to deliver my talk about my 5 years of PhD journey, what I have done so far. So I'm going to talk about unraveling multifaceted functions of novel ubiquitin ligase from Arabidopsis thaliana. I'm pretty sure that you must have heard the name of this model organism. It's a well-known model organism. So, okay, can you go ahead? So, a brief introduction before I finish showing you the data. So, plant growth and development are regulated by hormones and modulated by the hormones. So, from the time of the seed germination until they die, the growth and development of plants are regulated by intrinsic factors, and which are regulated by plant hormone. I'm pretty sure you know the names here, their function, and the environmental variables like drought, heat, cold, and salinity. And there is a balance, there is a crosstalk between plant hormones and the environmental variables, which ultimately guides how the guides and shapes the plant growth and development. So, in most of my slides, you will be hearing about the plant hormone. So, the gene I'm working with is a ubiquitin ligase. So, it's a part of ubiquitin proteasome system. There are two main evolutionary conserved pathways among all the eukaryotes. One is autophagy and second is ubiquitin proteasome system. Ubiquitin proteasome system is very vital for plant growth and development, and it's one of the most important cellular machinery required for protein integration. And it is the one that controls the process of ubiquitination. What happens in the process of ubiquitination? Okay, basically the ubiquitin proteasome system is composed of three main ubiquitin conjugating enzymes, E1, E2, and E3, and then a highly conserved globular protein called ubiquitin, which is conserved among all the eukaryotes, and then an ATP-dependent proteolysis complex called 26S proteasome. So in the process of ubiquitination, E1 activation enzyme activates ubiquitin at the expense of ATP, and then this charged ubiquitin molecule is transferred to the E2 ubiquitin conjugating enzyme, and then the gene I'm working is an E3 ubiquitin enzyme. So E3 ubiquitin enzyme, it picks up the ubiquitin molecule from E2, and also it simultaneously binds with the substrate protein. It picks the substrate protein and it transfers the ubiquitin molecule to the substrate, leading to the monoubiquitination. Now this monoubiquitinated protein is further expanded by the E3 like activity of the E3 ubiquitin ligases, leading to the polyubiquitinated substrate. In all the processes so far known, the polyubiquitinated protein substrate undergoes proteolysis using this 26S proteasome, and the protein is ultimately degraded. So, ubiquitin proteasome system in plants is required during stressful and under normal conditions for degrading the protein to create a platform for further signaling cascade and further signaling pathways. So, in a very, okay, I'm going to talk very little about plant E3 ligases. Mainly you can classify plant E3 ligases into two main categories: single subunit based egg, ring, and U-box. My protein is U-box protein, and multiple subunit containing SCF and SCF. These E3 ubiquitin ligases, they regulate a lot of cellular processes in plants, in animals, everywhere, because protein degradation is a must everywhere. Just to give you insight, Arabidopsis genome encodes 1500 there are some 25,000 proteins in Arabidopsis, also a lot in humans, and in humans 600. Yeast and cereals, these are the model of the 171. As you can see, there are a big plethora of E3 ligases in Arabidopsis because plants cannot move. They are sessile organisms, unlike animals, they don't move, and they need to maintain their proteolysis by standing at one place. So they have a lot of protein E3 ligases which target a lot of proteins, and they have to break things very fast. Okay. So there are some 64 plant U-box E3 ligases in plant out of those 1500, and a lot of E3 ligases have been uncovered for their functions. Two of the E3 ligases, 46 and 48, are known to be regulating the drought responses and oxidative stress responses. They have been their functions have been uncovered in our lab. So when I joined Professor Lugi Balzui's lab at BenGurion University, he gave me a totally new gene. Can we go next a little bit? Okay, can we go back? Okay, no, go ahead. One more. No, we are going back. Yeah. So he came to me and he said, there is a plant U-box E3 ligase POP41 in our lab, and there are some preliminary results available. Now we have to work on it and find out what this gene does. And that is how my journey in plant microbiology began. So I started uncovering what this gene does. So there was nothing much known about this gene, and there are some transcriptomic databases. And when I started digging those databases, I found a few things. Like here you could see the root of the plant. This shows highest expression of POP41. Then there was another chip sequencing database. Looking at that, I found that POP41 is a direct target of transcription factor called JACTA. And JACTA is it has been studied well, and it shows that JACTA is required for the root architecture development, and also it is involved in the auxin signal. So this gave me a hint where to look for the function of this gene, and I started focusing on the root. So my PhD composed of three main parts. One is studying its role in interaction with 1433. This was the preliminary results available in our lab. Second is POP41 and its role in development, and third is its role in the biochemistry. So I will go one by one and I will show you my data. Yeah, so I'm going to discuss about role of POP41 in its interaction with 1433 proteins. So the master student in our lab, he is the one who started working first with the gene, and he did this pull down assay. What is this pull down assay? You express the gene along with tag, JP, GST, whatever tag you want in plant, and you use the antibody against this tag, remove the garbage, you wash it, and then you send the sample for mass spectroscopy to see which are the protein targets interacting with this your gene of interest or the protein of interest. And among all the proteins, there were around 13. There are total 13 1433 proteins in plants, and all of them were found to be interacting with this protein, giving a strong hint that POP41 probably it interacts with 1433 proteins. So what are these 1433 proteins? So 1433 proteins are general regulatory factors. They are found in humans. They are also involved in cancers, and they also regulate cell cycles. They are found in yeast, and they are also present in plants. They are highly conserved protein, and they perform a large number of functions. They can bind, they can bond to the proteins, and they can change the subcellular localization. They can take a protein to nucleus, and from nucleus to cytosol. They can also change the structure of protein, and the important thing about these proteins is that these proteins requires presence of a phosphopeptide motif on their target protein to bind with the target protein. Next. With the binding chromatin analysis of the POP41 sequence, we found that POP41 contains two phosphopeptide motifs where serine and threonine are generally the target of phosphorylation. So serine 17 and serine 195 was the target. And we, in our lab, we did the mutagenesis, side-directed mutagenesis. We replaced serine 17 and 195, and we found that when you mutate the serine 195, the POP41 cannot bind with 1433. The serine 17 does not affect interaction with 1433, telling us that serine 195 is crucial for the 1433. So, because in our lab we did in planta pull down, so my next goal was to check the in vitro pull down is this reflects a direct protein interaction. So I purified 1433 is that protein from the bacteria. I did the bacterial expression, and per41 GFP was expressed in plant supernatant. And I did this in vitro pull down assay. As you can see in the second right hand line, when I pulled down per41 in the supernatant using the 1433 ligase, which was bound to the nickel beads, per41 was found to be binding to the 1433, suggesting that there is a direct interaction between per41 and 1433. So let me go next. So since we have proved that 1433 interacts with per41 protein, now what is the physiological significance of this interaction? For that, I have created phosphomimetic and phosphorylation mutants. What are these mutants? You replace serine with glutamine. Now glutamine, it mimics like a phosphoserine, like a charged phosphate residue. So this type of per41, it is like all the time it's always on. And then there is another one, phosphorylation null, you replace glutamine with alanine. So now this place cannot be phosphorylated. So it's like all the time it's off form. And now these form of per41, I have known under the per41 promoter. They have transformed, I have created transgenic lines, homozygous lines. And I have selected them, I have done the antibiotic selection, that's how you select the plant. It takes some 3 to 4 months to get, more than 4 months, 6 months to get a homozygous line. And I'm going to do tolerance assay and other types of assay to see what exactly, how the function and physiology of plants are modulated using these type of mutants. Okay, so I'm going to now the major part of my PhD includes understanding the role of per41 in its development, in the plant development. So how I began, how I started to understand where exactly per41 is localized and what it might be doing. For that, I created Gus transgenic lines of plant. What we do here is that Gus is a gene from bacteria. It's called glucuronidase enzyme, and glucuronidase enzyme. This enzyme cleaves the X-gal. It's a substrate, chemical substrate, and it produces a blue color chromogenic substance. So wherever you see the blue color, this is the place where the gene activity or the promoter of the gene is active. So I cloned this Gus gene under the control of per41 90 promoter, and I transformed the gene and I selected the homologous line. So as you can see in the A image, all the blue regions, it shows the gene activity. So I found that per41 is active in the reproductive tissues, mainly in the floral excision zones. And what are these floral excision zones like marked by arrows? These are the part where anthers and the floral parts are attached, and where the seeds are attached. So this indirectly suggests that per41 might be having a role in seed dehiscence and in the rupturing of the seedling. Thus the seed disperses. And another observations I made that per41 activity was highly observed in the root hairs. I'm sure you all know what are the root hairs. They are single cell projections. They are required for the absorption of water and nutrients for the plant. And also the per41 activity was present in the trichomes seeds. And these both plant organs are controlled by similar genetic pathways. And also in E you can see they are present, the per41 activity is present in the mature root hairs. And I would request you to focus on D image. The per41 is present throughout the root except this zone, it's called elongation zone. And to check next, like transcriptomic studies, I found that per41 is mostly expressed in the root tissue. So I took the root and shoot part of the plant and I did RT-PCR analysis. And as you can see in the last bit of the RT-PCR data, I found that indeed per41 is highly expressed in the root tissue and compared to the shoot tissue. Now we all know that ubiquitin proteasome system is active in nucleus and in cytoplasm. But sometimes the E3 ligase can be restricted to cytoplasm or nucleus. So to check, to understand where exactly the protein might be functioning or its target might be localized, I used this on the plants expressing puffer-T1 with GFP tag, and I did the focal microscopy, and as you can see in the bead, I found that puffer-T1 is present both in cytoplasm and in nucleus. And unlike animal cells, plant cells have a big vacuole between the segment. So that's why you can see that GFP is localized mainly at the periphery of the cells. And also further, I found, looking at the plants, I found that puffer-T1 is, the promoter activity was strongly induced in those small insect sections. You can see those are hydrozoans. Hydrozoans are the primary site of auxin synthesis. And later on you will learn that most of my work is related to the understanding the role of puffer-T1 and its interaction with auxin hormone. Also, it was present in the stomatas. And furthermore, I found that not only the promoter activity, but the transgenic lines expressing puffer-T1 GFP tag, the protein was accumulating in the same regions, suggesting that the role of puffer-T1 in hydrozoa development and stomata development. I found a lot of, during the journey I found a lot of hints and I gathered a lot of data, but I had to focus somewhere. So I continue in the role phenotype. Can we do next? So, to do the functional analysis of this gene, I started using mutants of puffer-T1. So for that, we ordered the mutants of puffer-T1, and 2 mutants we got from ADRC, Stock Center, Ohio. They provided mutants of C. And the 2 mutants are going to call puffer-T1 1 and 2. Puffer-T1 1 mutant has the tDNA insertion in the gene protein encoding region, and the second one has right in the UTR region of the 5' UTR. I did the bigger. Initially, I did the characterization, like to check if the tDNA is there or not. tDNA is some 5,000 base pair sequence, and it disrupts the whole gene. And how we make it, I will show you in the next slide. So we check through the PCR, and I also edit. Basically, to check if the gene is expressing or not, the best way is to check through RT-PCR. And like you can see in the image C, the puffer-T1 mutants were expressing very low amount of puffer-T1, which is like not significant, basically. Okay, can we go next? So this is how we make tDNA insertion. You take Agrobacterium tumefaciens. It's a naturally occurring soil bacteria, and you transform the gene of interest using a plant vector, and you put this gene between the right border and the left border. These are 25 base pair sequences, and the bacteria is expert in cleaving these 2 regions, and it will insert the whole cassette into the plant genome. And then we further keep selecting these seeds on the antibiotic plate, and you get the homozygous lines. So this is the first observation I made. So looking at the roots of puffer-T1 mutants, puffer-T1 1 and 2, compared to the wild type, you can see that the mutants show they show a reduced root hair phenotype in terms of elongation and in terms of density. Also, there was another phenotype which I observed was the lateral root density. Lateral roots are the secondary roots; they grow at the primary root. They grow perpendicular to the primary root. And I noticed that the whole root architecture was affected in the mutants. The primary root length was also reduced, and also the lateral root density was reduced. So it's well studied in plant sciences that auxin is the hormone that regulates the lateral root phenotype and also the root hair phenotype. As you can see here, the auxin maxima is required throughout the lateral root emergence. And in the root hairs, when you supply the exogenous auxin, it triggers the formation of root hair. And the concentration of auxin is maintained by the spatial localization of pin proteins. They are very important proteins in plant sciences, and they are well studied, and their roles are mainly for lateral root and the root hair. So to check if my mutants are not impaired in the auxin sensing, what if you give auxin to the plants and they cannot sense it, they cannot know it, auxin is around the plant. So to check that, I transferred 7-day-old plants onto the auxin arrays, and these above are control roots, and when you transfer to the auxin, you see the auxin triggers lateral root development. And what I found, there was no significant difference between wild type and mutant, and also I took the overexpression, where the puffer-T1 is constantly overexpressed. There was no significant difference, but there was the auxin was able to trigger the lateral root phenotype, suggesting that auxin sensing was not impaired in any of these lines. Can you do that? But here you could see when I looked at the root hair phenotype, when I treated for overnight, these are plants, same plants without auxin, and those are when I transferred into the auxin place. This is the study by Lee et al. And they show when you transfer plants with auxin, there is the auxin triggers the root hair formation. Only the overexpressor lines, where the POF41 is some 25 times expressing higher, they show a very high induction of the root hair phenotype, especially in the elongation zone. While this was not observed in the mutants. Can we do next? So to check if the mutants, because they lack the POF41 gene and the protein, to check if overexpressing POF41 can rescue this mutant phenotype, I took these, I gave these overexpression lines, and like you saw earlier, that mutants they lack the root hair, the density and the elongation. Here you see OE1, 2, 3, these are overexpression lines where POF41 is expressing in very high amount. And these plants, they express a lot of root hairs in numbers and intensity, even more than the wild type plants. And when you do the confocal microscopy, the D image, you can see the protein is highly accumulating in the root hairs, indicating that POF41 has to do something with the root hairs. But what happened in the lateral root? When using the same plant, when I look at the lateral root phenotype, the lateral root density, it did not rescue the lateral root phenotype. It further suppressed the lateral root phenotype and even the primary root. Now I tell you one thing, the lateral roots, they emerge from the pericycle cells, and the root hairs, they emerge from the epidermal cells and close to the primary root. So these are two different regions. The auxin concentrations, they fluctuate throughout the roots, and also they originate from two different kind of cells. So probably my gene could be playing two different functions. At one place it can be having suppressing activity, and at one place it might be inducing the root hair. So these are the plus plants. In control you see, the promoter activity of POF41 is minimal, but when you transfer to the auxin, like you saw, the root hair phenotype is too much in the overexpression line. When I transfer to the auxin, you see a very high increase in promoter activity of POF41 in the elongation zone where you saw the root hair, indicating that this is the place where POF41 activity is too high. Probably it's controlling the auxin concentration here, and that's why the root hair phenotype is too much here. And okay, next to check if the gene level, the mRNA levels are triggered by auxin or not. For that, I took the wild type plant and I treated them with the auxin. And as you can see, the B image doing the RT-PCR analysis using the primers against POF41. The POF41 levels, it went up after 7 hours treatment, like twofold. And also the protein level, these are the confocal microscopy images. You can see treatment with the auxin, it triggers the POF41 protein accumulation in the transition and the elongation. And another interesting feature I noticed during the confocal microscopy is that POF41 protein localizes at the apical and basal cell borders of the trichoblast cells. Trichoblast cells are the one that produces the root hair. So this is very interesting pattern. One need to be very observant and to understand what exactly, what this protein might be doing. And when you look into the literature, the pink proteins are the one. Here you can see that image. Pink proteins are the one that are local, that show an apical and basal localization. And the spatiotemporal localization of pink proteins, it determines the flow of auxin from the shoot to root and then the lateral transport of auxin. Since my gene has been showing auxin related phenotypes and the localization looks more similar to the pink, the next thing I had to check if POF41 has to do something with the pink proteins. There are total 7 pin protein members, and pin2 is the one that has to do with, that has role with regulating the lateral root and the root hair. Can we do next? So we check if POF41 interacts with pin proteins. What I did, I got these pin2 and cherry expressing Arabidopsis plants, and I cross-pollinated with plants, with my plants expressing POF41. I took the pollen grains of POF41 expressing plant and I put it in the flowers of pin2 and cherry expressing plant, and I did this homozygous selection, 3-4 months process, and I look into the confocal microscope to check if the two proteins are expressing together or not. Can you go next? So, why I did this process? To check the co-localization. What is co-localization? You do the confocal microscopy and you check if two proteins are sharing the same space. If they are interacting, they will share the same space inside the cell. So what I found that puffer-T1 interacts in plant within the plant, it interacts with P2. So you can see image A. The first one is puffer-T1 GFP, second one is P2 MJD, and third one is the merge. Arrow shows you here that two proteins are co-localized, suggesting the first step that these two proteins are together. They are probably interacting. Now, but this is still because inside the cell, the spaces are like too tiny. You cannot be sure even they are together, even they look together in the microscope, they cannot be interacting. For that, to check further, I had to do in vitro pull down assay. What you do in vitro pull down assay? Okay, pin proteins are the membrane proteins. You cannot purify these membrane proteins in absence of detergent. And once you put the detergent, you kill the functional activity. But here, pin protein, it has a very large cytoplasmic group, and it's very crucial for the localization of pin protein, for its activity, and even for the subcellular, for the transportation of the pin protein. And a lot of studies, they have used this hydrophilic loop region. They express this hydrophilic loop region, and this you can use to check the in vitro binding assay. So I expressed this protein, P2 hydrophilic group in bacteria, verified it. It's a 61 kilodalton protein, and I purified puffer-T1 GFP in the plant. Supernatant, I added this P2 hydrophilic group in plant supernatant, where the puffer-T1 GFP was expressing, and you do this 1 hour, 2 hour rotation on 4 degrees Celsius, and then you use antibody against the P2 GST. Now, if puffer-T1 is binding with P2 GST, it will come down when you will pull down using the GST. You run over the gel, and as you can see in the image C, puffer-T1 GSP was found to be interacting only with the P2 GST, not with only GST type used as a control, and not with the GSP type. And another very interesting feature I observed in these plants was when I did the focal microscopy. These are the wild type background of the plant. The first one, here you see only P2 MJD expressing in the wild type plant. And these plants are the one where P2 MJD is expressing in the background of puffer-T1 GFP. Puffer-T1 is an E3 ligase. So probably it is doing degradation of some repressor that suppresses the expression of P2 up to the elongation zone. But when you express P2 protein in presence of puffer-T1, the expression goes further up, suggesting that puffer-T1 probably stabilizes the P2 protein. And like these images I have been showing you since the beginning, these are the zones I noticed in the beginning of my research, and by the end, it makes sense that puffer-T1 activity is more here in this zone, and also auxin triggers puffer-T1 expression more in this zone. So probably when you express P2 protein in presence of puffer-T1, this puffer-T1 expands and the expression of P2 by stabilizing this. Okay, so now since everything has to do with the auxin, I don't know what are the intracellular auxin hormone levels, and when I see that puffer-T1 levels are going up in the elongation zone, I want to see what is happening to the auxin concentration. And for that, I have created the reporter line where you can visualize the auxin. Auxin is a very small, it's kind of an very small molecule. To check the auxin level in plant, I have created transgenic lines where you can express the TD tomato tag, the red tag here in the nuclei of the cell, and it has the MT2 domain. What does this MT2 domain does? If you have high auxin concentration in certain parts, the protein won't degrade. But if you have low auxin concentration, the protein will degrade. So like I showed you, the research many research have showed that high auxin concentration is required in the lateral root energy. So there you see lateral root, and you see you have a high auxin concentration there, and that's why the protein is stable there. But rest of the root part, it has very low oxygen concentration, and there the protein will be degraded. So I am going to study further what exactly is happening to the oxygen concentration to understand the background. Okay, now I'm reaching to the third part of my research, which is to see the role of copper T1 in Arabidopsis. So this was the preliminary study done in our lab, which I further continued, and I found that copper T1 mutants are drought hypersensitive. What is, I guess you all know what is drought. Drought, when I exposed these mutant plants during 1952 using the mutant of copper T1, and when I stopped giving waters to the plant, these were the first plant to die of suggesting that if you overexpress this protein or if you stabilize this protein somehow, probably in future this will help in making plants drought resistant. Then we go next. And the second observation and second study I did was to check the oxidative stress response in buffer-tube mutant and overexpressing plant. So these are the Gus lines, and when I incubated these plants with H2O2, the promoter activity was suppressed in the primary root, and the next, like when I germinated these plants in presence of H2O2, you can see the mutants, they showed more like a resilient phenotype to the oxidative stress, unlike the wild type plants, suggesting that PUF1 PUF1 is negatively regulated by the oxidative stress. Also, the RT-PCR result shows an increase in PUF1 RNA when treated with hydrogen peroxide. And when you look at the protein localization, it showed a very interesting pattern. These are the confocal microscopy images. And when you treat the plants for 2 hours in hydrogen peroxide, very less concentration of 5 millimolar, the primary root shows a suppression in the protein levels, and the elongation zone where the root hairs emerge, it shows very reduced protein levels, but the upper root region, it shows a slight increase in the protein levels, and also the protein localization, it changes in response to the hydrogen peroxide. The protein goes to localize on the apical and basal borders. These are the mainly regions where the active sites for the ROS metabolism applies. Can we go to the next? And another other abiotic stressors I checked were ABA, which mainly regulates the stomatal closure to reach out, and the NaCl, I treated plants and I did the RT-PCR analysis to check what happens to their mRNA levels. ABA and NaCl, they positively regulate the mRNA levels. And in terms of heat, this was a very interesting phenotype I noted. When you treat plant for 1 hour, you do this microscopy, the protein forms punctate structures, like they form sphincter, which has been seen in autophagy. And there are not many known E3 ligases which play a role in both autophagy and ubiquitin proteasome system. So this will be my next goal, to check if my PUF1 gene is also taking part in autophagy. And there was also the stress where the protein levels were completely suppressed. Like in 1 hour, you can see 45 degrees Celsius treatment, the protein was reduced to a high extent, and when you do 2 hour treatment, protein completely vanishes. And also the mRNA levels goes down. So this suggests that PUF1 is negatively regulated by the heat stress, and we'll check further in what happens in the overexpression lines. Can you go next? I'm going to skip these slides because this is just the preliminary data. Okay. Can you go next? Yeah. So I would like to thank my lab where I'm currently working, Professor and all the lab members, to have Dr. thaliana, and also I would like to thank you, all of you, for coming here and